How did patients fare in the GALE extension study, which assessed the safety and efficacy of Pegcetacoplan for GA?
I'm Scott Griswanis here with Greg Notstein, and this is New Retina Radio from Retina Today and Bryn Mawr Communications. Dr. Nathan Steinle shares the results of the first 6 months of GALE, in which patients who received treatment for 24 months in Derby and Oaks continued with therapy, and in which patients who had received sham injections for 24 months were switched over to treatment.
And Dr. Paul Hahn joined us in our mobile studio in Seattle. He shared data from a matching adjusted indirect comparison of Pegcetacoplan and Avacincaptad Pegol. What did he and his team find? Stick here to find out.
Pegcetacoplan was the first treatment approved for geographic atrophy in the United States. The Phase 3 Oaks and Derby data showed us that Pegcetacoplan therapy was most distinct from sham therapy during months 18 to 24.
Did the trends seen after months 18 to 24 continue with further treatment? Dr. Nathan Steinle has the answers. Dr. Steinle practices at California Retina Consultants and Retina Consultants of America in Santa Barbara, California. Dr. Steinle, welcome back to New Retina Radio.
Oh, it's a pleasure to be here. Thank you for having me.
We've spoken about Derby and Oaks on this podcast plenty of times, so no need to go too deep into the design here, but give us a quick refresher and then tell us about the GALE study.
Absolutely. So Oaks and Derby were the Phase 3 trials for Pegcetacoplan that allowed it to be registered in February of 2023 as the first on-label treatment for geographic atrophy. And these were divided into monthly, every other month, and sham doses and followed out to 2 years. At that 2-year mark, patients then can enroll over into a long-term extension trial that's called GALE, another horse term, G-A-L-E, GALE. And so what this is a 3-year observation study where we are actually going to see all patients receive active treatment. So those patients that were receiving monthly therapy in Oaks and Derby continue on monthly therapy. Those receiving EOM or every other month therapy in Oaks and Derby continue that treatment in the GALE extension trial. However, the more interesting part is those patients that got sham monthly or sham EOM in Oaks and Derby now are getting active treatment at that same regimen going forward for GALE. And then GALE will be a 3-year extension trial. And we presented recently the 30-month data cut. So we have now an extra 6 months of data on these patients. And overall, it's really interesting. We had a very large database for Oaks and Derby, and 83% of patients, which is about 800 patients, rolled over into this long-term extension trial. So we're going to have a really good snapshot at how Pegcetacoplan works over time. Right now, we have a 30-month data cut, and at the end of the GALE trial, we'll have a 5-year data cut on these patients.
So you just talked about how many patients actually stuck around and enrolled in this extension study. About how many injections were given?
Great question. So at the 30-month mark, we had 15,000 injections now of Pegcetacoplan. And as GALE now continues to move forward, it's pretty impressive. We now, overall in the entire clinical program between Philly, Oaks, Derby, and GALE, we have 23,000 injections of Pegcetacoplan. So it's a molecule we've been working with for many years, since about 2018. And it's exciting now to have, these almost 800 patients now rolling forward into GALE to give us even more data going forward.
Okay, let's start with the continuous treatment group. What did you find after 30 months of Pegcetacoplan therapy?
Really good question. So you can kind of divide this into two categories. Those patients that had treatment in Oaks and Derby for 2 years, how did they do going forward? And the answer is they continue to do better than those patients would have done without treatment. And what I mean by that is there's more differentiation over time. So as Pegcetacoplan seems to work more and more and more over time. So if you look at that final 6 months data cut, now from month 24 to 30, there's a further reduction in GA progression, both in monthly and every month dosing. And what's more interesting now is that we finally now have crossover patients. So we have patients that had 2 years of close observation in the clinical trials of Oaks and Derby, where they're getting sham injections. And now they're rolling over to active treatment into GALE. And so we finally can say, what other signals do we have that this molecule really works for GA? Well, this is a great signal because we have crossover patients. And so in those patients that receive either monthly or every other month dosing, Now, in the first 6 months of this extension trial, GALE, we did see a reduction in GA progression in those patients. And it was on the exact same order, about 15% in the first 6 months. It was on the exact same order as these patients had a baseline across OAKS and Derby for the first 6 months of OAKS and Derby. So it seems to be very consistent.
You also looked at patients with only non-subfoveal lesions who received continuous treatment. What did they show?
Yeah, really great question. So if we break it down into extrafoveal lesions, and the criteria for an extrafoveal lesion was very, very broad. It was any lesion that did not involve the foveal center. So again, extrafoveal is any lesion that did not involve the foveal center. These patients did exceptionally well. So now at the 30-month mark, we had a 45% reduction in GA progression and a 33% reduction in GA progression in monthly and EOM dosing at that 30-month mark. So these patients with the extrafoveal baseline lesions seem to respond best to pegcetacoplan.
Safety is one of the biggest questions in GA therapy at the moment, so let's focus there for now. We'll start with new onset exudative AMD. What did your team observe in the GALE study?
That's a really great question. So what we saw in Philly, which is the phase 2 trials, was a little bit of a surprise to us, was this dose-dependent conversion from dry to wet AMD. And so when we looked at the phase 3 trials, OAKS and Derby, we again saw this dose-dependent conversion to wet AMD. And we see that again in GALE. So as patients had more therapy, whether it be monthly or EOM therapy, they had an increased rate of conversion to wet AMD. And those patients then that now have crossover are converting at a kind of the same rate to wet AMD. So it seems to be pretty consistent across the board.
And then what about endophthalmitis and intraocular inflammation events?
In GALE, the first 6 months of GALE, there was not a single episode of a infectious endophthalmitis. So now the overall rate across the study platform is 1 in 3,700 injections for endophthalmitis. And that's great news, because when we're talking about complement inhibition here, we're talking about, modulating the immune system. And so we were a little bit worried that we would see an increased rate of endophthalmitis when we started these trials, because we are, we're suppressing the immune system at baseline, and we're just not seeing that in the trial. So that's really good news. And your second question about the IOIs or intraocular inflammation, the overall rate was 0.26%, which is about 1 in 400 injections for IOIs. And most of these are non-serious and just a few serious IOIs.
Now, that said, there were some real-world reports of IOI that included retinal vasculitis. These were pretty serious. They were shared this year at the ASRS Annual Meeting. You commented on them in your presentation. Can you remind us what you said?
Yeah, great question, Scott. So what got a lot of press was the reports of vasculitis in the real world use. And, just to kind of give a baseline to this, we now have 23,000 injections in the trials across OAKS, Derby, Philly, and GALE. And we just haven't seen any occlusive vasculitis or vasculitis in these trials. And any of the patients that had an IOI or inflammation event in these trials, we went back and looked at and say, hey, maybe we missed the vasculitis, and we just didn't. And then we looked at all these patients, made sure they had FAs that had the serious IOIs, and even had expert, external people look at these images, uveitis specialists, retina specialists, and they agreed there was no signs of vasculitis. So we just haven't seen that in the trials. But then when you release a drug into the real world, that denominator gets bigger, and so they've had 68,000 vials shipped in the United States since February, and they think around 62,000 of those, 90% of those actually been used in eyes in the United States now. And so what I reported on in Seattle was 7 cases of vasculitis and occlusive vasculitis that have been reported to the sponsor company. And when we look at those, we see that 3 of those have now resolved, and 4 of those are indeed serious events. We think the overall incidence of this rare event is on the order of 1 in 10,000 injections to date, about 0.01%.
All right, now let's head back to the GALE study here. Can you give us a nice wrap up in summary to what you presented?
Yeah, the take-home message is this drug works. There are multiple different data points that we can look at to look at this. Number one, we finally have crossover patients, those patients that had 2 years of sham therapy in OAKS and Derby that now have active treatment in GALE, and we see that reduction in GA progression in those first 6 months of GALE. Number two is that we have continued differentiation of those patients who do receive active treatment over time. So this drug works more the longer you give it, which makes sense because you're kind of slowing down the aging process here. And so in the final 6 months here from 24 to 30, we showed in GALE, we see that further reduction both in subfoveal and especially in extrafoveal lesions across the use for pegcetacoplan.
Dr. Steinle, thank you so much for coming back on the show and sharing your research with us.
Hey, what an honor. Thank you for having me.
Hi, listeners. This is Scott. A quick production note. Greg and I recorded this conversation with Paul Hahn at ASRS in Seattle. That was before the approval of Avacincaptad Pegol by the FDA. You'll hear us referred to a pending or a forthcoming approval. Well, in the real world it's happened, but during the interview it hadn't happened yet. So keep that in mind while listening. Enjoy! What an unusual but exciting time to be a retina specialist. A disease, GA, for which we have had no approved agent, has finally seen a therapy cleared by the US FDA, and a second drug may be approved within months. Both of those drugs went through vigorous pivotal studies, data from which were submitted in their approval filings with regulatory bodies.
The studies, of course, did not compare themselves to one another, and no head-to-head study exists or is even recruiting. But is there something in the pivotal study data we can glean about the relative efficacy of these drugs? Dr. Paul Hahn thinks there might be. He's with us today in our studio here in Seattle during ASRS 2023.
Dr. Hahn practices with NJ Retina in Teaneck, New Jersey. Dr. Hahn, welcome to New Retina Radio.
Thank you. It's my pleasure to be here.
Pegcetacoplan and Avacincaptad Pegol are complement inhibitors delivered via intravitreal injection. The former is approved for GA, while the latter is awaiting word from the FDA. Those are the similarities. Tell us about some of the important differences between these two therapies.
Yeah, so pegcetacoplan and Avacincaptad Pegol are both complement inhibitors, as you mentioned. Now, pegcetacoplan targets a little bit further upstream, targeting C3 and C3b, whereas Avacincaptad Pegol targets a little bit further downstream towards something called C5. These are both different elements of the complement cascade. Their trial designs were different as well. Pegcetacoplan was studied in two parallel phase 3 trials called Derby and Oaks, including a broad range of patients, including both non-subfoveal as well as subfoveal lesions, and looking at monthly as well as every other month dosing. In contrast, Avacincaptad Pegol was studied in only a single phase 3 or true phase 3 clinical trial called GATHER2, as a GATHER1 study was a phase 2/3 study. This single phase 3 study, the GATHER2 study, looked only at non-subfoveal lesions and looked only at monthly dosing.
And the time endpoints for both of those studies were different, correct? In Derby Oaks, it was a longer study than GATHER2?
Their primary endpoints were at a year, but Derby and Oaks does have data out to 2 years.
Okay, so GATHER2 only looked at patients with non-subfoveal GA lesions, whereas Derby and Oaks each looked at patients with both non-subfoveal and subfoveal lesions. What happened if we crunched the numbers, combined the data from Derby and Oaks, and looked at only non-subfoveal patients? What were the findings?
Yeah, so that was a pre-specified endpoint in the clinical trials. And if we look at Derby and Oaks combined, we saw a 26% reduction in the monthly arm with pegcetacoplan and a 23% reduction in the every other month arm, and both of these were statistically significant. In GATHER2, we saw a 17% reduction with monthly Avacincaptad Pegol.
So just looking at non-subfoveal patients gives us some type of comparison, but you and your colleagues performed an anchored matching adjusted indirect comparison. Tell us more about that type of analysis.
These MAICs are somewhat new to us, but they are a well-validated approach, both in ophthalmology as well as in other fields. These MAICs are often sometimes required by regulatory agencies for approval as well. What these MAICs do is allow us to compare different trials in the absence of a head-to-head comparison. The approach for this MAIC is first based on matching patient populations between two different trials. So in two different trials with different inclusion and exclusion criteria, you would apply the more restrictive inclusion and exclusion criteria to the less restrictive trial. In the case of Derby and Oaks versus GATHER2, as we've talked about, GATHER2 had more restrictive criteria. They only looked at patients with non-subfoveal GA, and they only looked at patients with, they excluded patients with fellow eye CNV and included patients only with a certain range of visual acuities, in contrast to Derby and Oaks, which looked at both non-subfoveal and foveal GA, did not exclude patients with fellow eye CNV, etc. And so what we did was we took the patients at Derby and Oaks from an individual patient level and filtered out only those who had the same inclusion and exclusion criteria as GATHER2. So only the non-subfoveal patients, only those without fellow eye CNV, etc. Once you have this matched population, the next step is to try to balance those two populations among different pre-specified criteria. So you perform inverse propensity score weighting to create similar populations in terms of things like vision, lesion size, multifocality, bilaterality, etc., to create not just a matched population, but also a balanced population.
That is a lot of filtering, but we started with big populations in both studies. What types of populations did you end up with in Derby and Oaks after applying the GATHER2 inclusion-exclusion criteria?
So you're absolutely right. When you apply all that filtering, you're going to reduce the number of patients that you have. In our study, in the Pegcetacoplan monthly arm, we ended up with 61 treated patients in the Oaks trial and 49 patients in the Derby trial, with a sham arm of 42 in the Oaks trial and 53 in the Derby trial. For the Pegcetacoplan every other month assessment, we had 54 patients in Oaks treated with Pegcetacoplan and 42 treated with sham. And in Derby, we had 55 patients treated with Pegcetacoplan and 53 with sham.
You and your team looked at each Pegcetacoplan study versus GATHER2, and then you did a full meta-analysis. What was the outcome you used?
Our outcome was GA lesion area change in square millimeters from baseline to month 12 between Pegcetacoplan monthly versus Avacincaptad Pegol monthly and Pegcetacoplan every other month versus Avacincaptad Pegol monthly.
All right, that is a lot. So let's tackle the monthly comparisons first. We could get deep into the weeds here, but we're going to keep it top line. What did you find when comparing monthly Pegcetacoplan with monthly Avacincaptad Pegol?
Yeah, that monthly comparison is probably the most apples-to-apples comparison. And in comparing these, we looked at differences between different trials. We compared Oaks versus GATHER2, we compared Derby versus GATHER2, and we compared combination of Oaks and Derby versus GATHER2. In comparing Oaks versus GATHER2, we found a 37% greater reduction with Pegcetacoplan monthly compared to Avacincaptad Pegol monthly, and that difference was statistically significant. When we compared Derby versus GATHER2, we found a 12% greater reduction with Pegcetacoplan compared to Avacincaptad Pegol, but this difference was not found to be statistically significant. But when we combined Oaks and Derby and did a meta-analysis comparing Oaks and Derby combined versus the results in GATHER2, we found a 30% greater reduction favoring Pegcetacoplan monthly compared to Avacincaptad Pegol monthly, and this result was also statistically significant.
The next comparison is a little trickier. It's not quite apples to apples, to use your phrase. This is Pegcetacoplan every other month versus Avacincaptad Pegol monthly. Tell us what you found.
Well, the reason we did this was because Pegcetacoplan every other month was tested in the clinical trials, and we thought it would be useful to see how less frequent dosing with Pegcetacoplan might compare with more frequent monthly dosing with Avacincaptad Pegol. And what we found was that in each of the comparisons, Oaks versus GATHER2, Derby versus GATHER2, and combined Oaks and Derby versus GATHER2, we did not find a statistically significant difference between any of the comparisons, although numerically they all favored Pegcetacoplan. And what that suggests is equivalent efficacy between Pegcetacoplan every other month versus Avacincaptad Pegol monthly.
That's a lot of studies. It's a lot of data points. What are the top takeaways that you would provide to your colleagues?
A few. So first off, I think it's important for us to be aware of this MAIC approach. As more therapies become available, we're going to need methods to compare different drugs between different trials in the absence of head-to-head comparisons. From this study, we demonstrated that pegcetacoplan showed a better efficacy profile than Avacincaptad Pegol among patients with non-subfoveal GA. Specifically, that we saw a greater reduction in GA lesion growth at month 12 with monthly pegcetacoplan compared to Avacincaptad Pegol monthly. But even if we compared less frequent dosing of pegcetacoplan with every other month, we saw equivalent or comparable efficacy with Avacincaptad Pegol monthly.
Dr. Hahn, thank you so much for your hard work in this study, and thank you for joining us here in Seattle.
My pleasure as always. Good to be here.
That concludes our ASRS 2023 coverage on New Retina Radio. If you missed our first episode, go back in your feed and find it.
And for continued coverage of all things retina, be sure to rate, review, and subscribe on your favorite podcast app.